Okay, so everybody, thanks for the great talks. It was lots of good new stuff there. It was exciting. So I'm going to start off reading the questions here from the. And if I read your question and you want to clarify anything, it's okay to speak up. You know, you don't have to be anonymous. It's not the point of this. So if you want to clarify, you can have a direct dialogue with the speaker. We're not trying to turn you into the Snapchat, Instagram culture of your kids, where you don't talk to people face to face. Anyone who asked that question want to clarify anything? Okay. All right. So, Dr. Baumgarth, have you compared the CD4 T cell profiles of the C3H and B6 mice in the context of Borrelia infection?
No, we have not. But Dennis Weiss actually spent about 20 years making a recombinant inbred strain of C3H and C57 to identify the genetic basis for the difference in why C3H mice develop arthritis and black 6 mice do not. And the genetic basis she identified was a gene, an enzyme that organizes the glycosaminoglycan pathway. And effectively what happens in C3H mice, they accumulate these glycosaminoglycans, which acts as a pro-inflammatory agent, and that then induces an immune response and arthritis in these mice. So this really doesn't have anything to do with the immune system. That has to do with the activation of the immune system simply by this enzymatic defect that is specific to the C3H mice. So it's wonderful that she found it, but I think what it means for us is that there's really no underlying immunological basis for this difference.
Right. And also, if people have follow-on questions to questions that we ask, feel free to raise your hand. I mean, for a follow-on question, it's okay. All right, if no follow-on questions, Dr. Zhang, have you tried to exploit the use of RNA sequencing data to identify other drug targets?
That's a great question.
Or drug candidates,
in fact, I didn't have time to go into that. In fact, the RNA-seq analysis allowed us to identify a range of targets that are useful for not only for the treatment, but also for diagnosis. We know that the inclusion of the persistent antigens that seem to improve the sensitivity of diagnosis. And in fact, the CLIP-P protease is an interesting target. I think Kim Lewis has some interesting results where activating the protease seem to reduce the persisters. That's really known persistor target that can be potentially good therapeutic targets. And I don't doubt these potentially can serve as potential good targets for therapeutic as well as for diagnostics. Yeah.
All right, there's a follow-up question. Yeah, go ahead.
So you also found in your drug screens, you found some drugs that seem to work better on the persisters. Does that correlate in any way with your RNA-seq data? Like the targets of those drugs?
Yeah, right. Okay, right. That's a good question. No, in fact, not. So the ones we found to be good active against Borrelia persisters, these primarily act on the membrane. Very interestingly, like daptomycin, clofazimine, essential oils, amazing activity. They seem to act on the membrane. So in the persistor field, people know that drugs targeting the membrane seem to be good at killing persistor bacteria. And also drugs that damage DNA that appear to have preferential activity or good activity killing persistor bacteria as well. Yeah.
Yeah, one more follow-up question there. Yeah.
So I actually next event but I've got multiple translations. So Nicole, I wanted to ask you, do you have any idea how Borrelia corrupts the CD4 cell?
Well, I have that question here. I just haven't gotten to it. Oh, there's a follow-on to Dr. Zhang's. Yeah, it's okay. That's okay. That's fine. We'll go right to that question. Yeah. So, so this is for Dr. Baumgarth. What is the mechanism by which you think Borrelia suppresses or corrupts the CD4 cells?
I don't know. I wish I knew. I think what You know, I'm an immunologist, so what we are doing is we're looking at the targets of the Borrelia-mediated immune evasion as we understand it. And it would be wonderful if we had some microbiologists who would work on the other side to identify the genes in Borrelia that could actually facilitate that. As you know better than I do, Borrelia is not known to secrete anything actively. So however, what we have shown is that Borrelia is directly in the lymph nodes, which is where these CD4 T cells are activated. So there's the potential of these cells, either directly or indirectly, to interact with the CD4. So we really, so the data, and I don't want to go too much into the sort of nitty-gritties of it, but from what we are looking at, it seems like the first phase of the activation of the T cells happens, but then that second part that sort of determines what their quality is and their functionality, that seems to be the part that isn't, I wouldn't say not working, because we haven't done enough work to demonstrate that, but there's something about that second part that's not happening, whether that is directly because the Borrelia interacts in some way specifically with the CD4s, or indirectly in the way that Borrelia interacts with the tissue, the lymph node itself, and how it changes the structure of the lymph node is something we have to look at.
Yeah, go ahead.
That all makes sense. Just, unless you're even microbiologist, would try out, because you would have to do a thousand experiments. It's not that. I would rather suggest a biochemical approach, fractionation and do a biased purification. Something we do routine.
Yep.
Great. We'll get a microphone out there eventually for people to speak here, so you can hear. I'm sorry, it's hard to hear. So this is for Dr. Embers. Does Borrelia have a quorum sensing system, for example, homolactoserine, that allows it to recognize a component of the spent media?
Yes.
Yeah.
No, it's still not.
Maybe it's yours is not working. Yeah, I'll borrow that one.
Borrelia, yes, Borrelia does have a quorum sensing system, and they probably do produce these autoinducers. Goes through the LuxS gene product, and so when we've looked at VBNCs, I think if we look at gene expression, we're going to see indications that the quorum sensing is affected. So.
Question for Dr. Zhang. Could you elaborate a bit more on the arthritis model you've created?
Yeah, so the arthritis model is done with Borrelia strain N40, which is in vivo passaged. It's very interesting, depending on, you can use different forms. In this particular study, we're actually evaluating different forms of Borrelia, where we have a log phase culture, we have a microcolony form of the culture. Interestingly, the biofilm microcolony form of the bacteria seem to be more pathogenic, causes arthritis much more quickly than the log phase form of the. So we think this could be just like, this is why we propose this kind of a type 1 PTLDS, type 2 PTLDS. That is during the very early, very initial infection stage. If you used a persister form, then you would actually cause a more persistent form of the disease. That we think would be a good model for evaluating drug combinations.
Follow on right there? Yeah.
So with that model where you're using
we actually have a microphone.
Yeah, there we go.
where you're using that model, using different forms of Borrelia to initiate the inflammation in the joint, do you have some way of quantifying the amount of spirochetes going in? Because obviously it's easier to enumerate spirochetes when they're free form versus in a clump.
Right. We actually use the same inoculum size. We actually we counted during the infection process. We use this planktonic form versus this microcolony form. Yeah.
Okay. Follow-ons? Okay. Okay, Dr. Baumgarth, Your data shows an increase in FOXP3 positive Tregs. How might these cells be contributing to the dysregulation of the TFH? Might they also be a therapeutic target?
How the Tregs might interfere with the Tregs?
Yeah, so if whoever asked this question maybe could clarify. Yeah, there we go.
Yeah, you showed in your pie chart
Yeah.
that FOXP3 expressing cells were kind of a big part of the
Yes. So when we did the study initially, we compared a difference. When we initially looked for FOXP3, sort of the question of whether Tregs could just be there, a lot of them, and they would suppress the T cell responses. We compared that to immunization. We didn't really see a big difference. And so the piece of data that we don't yet have is whether any of those Tregs are T follicular regulatory cells, so the subset that actually interacts in the B cell follicle. And that would, tell us whether there's an interaction. I think, so I didn't want to make too much of this Treg. I think rather than there are too many Tregs, there may be too little effectors. And that may look like this big bias away from, or this sort of big pie of the Tregs. Not all our data suggests there are too many Tregs, so I think it's more like too little of the other guys. But we'll have to do those studies.
Okay. All right, Dr. Embers, what do you think triggers the VBNC state in the host?
So when I talked about the natural induction of the VBNC state, I think that's probably what this question is about. In primate hosts, the level of Borrelia is extremely low. It's a very low level infection, whether antibiotics are used or not. So I think, and you also find them in these immune privileged sites and in collagenous tissues, and they're in areas typically that have low nutrient content, and they're also evading the immune response. And so I think that the combination of the site and the immune evasion phenotype are what drives the quiescence of Borrelia in primate hosts.
Want to follow on that? Okay. Okay, no further questions about that. All right, Dr. Zhang, can you speak more about the essential oils disrupting the cell membrane? How do the essential oils compare to the persister drugs? Will evaluating the oils be part of your studies going forward?
Yes, that's a great question. In fact, I get more inquiries about the essential work. So it's really very interesting. We didn't expect, we thought essential oil just for aromatic purposes, and then when we tested some of them, it's really quite amazing. The activity is really even better than the drugs we found to be active, like daptomycin. At 40 micromolar daptomycin, the essential oils seem to be more active than the daptomycin, so which is really very surprising. But of course, it's actually used in the context of a mixture. So that's why oregano oil, it's actually the active component is carvacrol. Oregano oil coming from different parts, having different percentage of carvacrol, with the ones that containing higher percentage of carvacrol showing better activity. Of course, this actually also shown, seemed to be the case in patients, because I just been to France, attending their Lyme conference, where I heard physicians getting their oregano oil from Greece, where it's a source that contains 80 to 90% of carvacrol, which is quite amazing. Whereas they know that is oregano oil containing less percentage of carvacrol seem to be less effective. So they have a lot of accumulated a lot of experience already. So I think it's really very interesting to evaluate these essential oils alone as well as in combination with antibiotics, which I didn't have time to go into that. This really has also activity on dissolving biofilms and also making biofilm structure loose as well as allowing perhaps other drugs to go through the membrane more easily as well. So that's why there's a potential to use it in combination with antibiotics as well. So I think there's a lot of potential opportunity to, I think a lot of potential to finding essential oil in curing or in not curing I don't know, we are allowed to use the word cure here, because when you talk to Dr. Horowitz, he never says the cure, right? He always says putting patients into remission, right? So I think it's really an interesting mix with essential oil and antibiotics to further evaluate in our animal models first, as well as doing clinical trials. Yeah, thank you.
Any follow-on questions on the essential oils? No? There's one right there. No. You got a microphone coming for you, sorry. I want everybody else to hear.
So I assume you would give this orally to the animals. Do you think that the bioavailability of these oils would be?
Yeah, that's a great point. So that's exactly what we're working on, trying to figure out the PK, pharmacokinetic, the toxicity issue, and the dosing schedule, how often we use, as well as in what form of the oil, whether you can't really quite use it directly, you have to somehow put it in a gel capsule or mix it with food, as well as mix it with carrier oil. That's make it more tolerable. But there's more work to be done on that to find out the best dosing schedule and to use it safely and effectively.
All right, thanks. Dr. Baumgarth, have you looked to see if Pd1 levels are altered upon Borrelia infection in different tissues?
No, we haven't done that. We are focused right now on the lymph node and on the expression of PD1 on CD4 T cells. So we would like to look in the skin now, having seen these effects and maybe some other sites, but we haven't yet looked.
Okay. All right. Dr. Embers, what is the significance of culturable VBNC to the wider Lyme community? Does it refute Dr. Wormser's work? What more needs to be done?
I like this. So I think it's very important because it not only proves that the spirochetes are viable, but it would offer an opportunity to, which we haven't done this yet, but it would offer an opportunity to resuscitate them after antibiotic treatment and then determine their infectivity. And I suppose that they would probably remain infectious. So I think that it offers not only a means to show that the spirochetes are viable after doxycycline treatment, but also to improve our efforts in determining the efficacy of treatment and potentially these could be used with human cultures, skin biopsy or blood cultures.
Any follow-on questions for that? No? Yeah, one run back there. All right.
Monica, it's not really a follow-up question, but a comment. I think another really important aspect of the work, if it turns out this way, is with a larger number of animals being tested, that you would potentially find some non-human primates responding favorably to a doxycycline treatment, and you will not be able to recover organisms after treatment, but then in other animals you will. And I think that would also better recapitulate what happens in the human Lyme patient population. There are some individuals where a combination of standard of care antibiotic treatment, 10 to 21 days of doxycycline, and their immune response is sufficient to kill and clear the organisms, and they don't have any persistent symptomatology. But then there are others, obviously, that don't follow that idealized path.
Yeah, we've not yet had the opportunity to use these refined culture medias in non-human primates, but I'm very excited to try this. And I think, yeah, it takes away the argument that they're not viable from that contingent. And so I'm not saying that it's going to work with humans, but I think it's certainly worth a try.
All right. Okay, Dr. Baumgarth, what is the next step for interrogating these sort of enigmatic TFH cells? Are you planning something like single cell or cytoph to see if they're part of a bigger puzzle in the, or picture of the immune response?
Yeah, I mean, I'm really interested in how, why the germinal center response collapses. So this is where you make high affinity antibodies. This is where what I really would like to do is reverse the collapse of the germinal center and to see whether if that collapse doesn't happen, whether we could achieve clearance with antibodies. Because what I've shown you is that IgG is really important in controlling Borrelia load. And so the question is, why can you get rid of 99% of the Borrelia with the antibodies, but not 100%? And so one approach would be to overexpress BCL6 and see whether that will actually change the immune response and the outcome. And in terms of the mechanism of induction of BCL6, our first step is obviously repeating this and then seeing exactly where this defect is and what causes this defect, because when you activate CD4 T cells, the first thing they do is upregulate BCL6. All the T cells do that independent of what becomes of them later on. And we see some BCL6 expression. It seems that first step happens, and then the second part, the sort of more long-lasting induction of BCL6, seems to be when the T cell interact with the B cell, and that's where we don't seem to be seeing that. So we would like to probe that further. In terms of what techniques to use to do that, yeah, we'll throw everything at it that we have. We don't have a site-off on campus, but you know, I'm not sure we need 50 color flow cytometry. We can do 20 colors, so that might be enough.
Great. All right, Dr. Embers, would your results be expected to be any different using a bactericidal antibiotic?
Yes, I think it's certainly possible. I didn't talk about this, but we did send our media to this lady, and she used injectable ceftriaxone, right, to treat the mice, and it was a different and harsher treatment regimen, and they did not grow. However, we don't have PCR data for those to determine if they were eradicated or not. So I didn't talk about it. But in our hands, I think, and in their hands, that the injectable ceftriaxone is significantly more effective.
Yeah, if I could just clarify. So the way we did it was ceftriaxone twice a day for 5 days, and then once a day for 25 days, which is a lot for these little mice. So, and then I think we waited 1 month before we took the tissues. And that we chose that regimen because that's what Barthold showed in the studies that showed that after 12 months he could find some PCR positivity, but not after 1 month, 3 months, or 6 months. So at that time where we couldn't culture them out, there was also a time where Barthold never saw any PCR positivity, and we cultured some of the tissues. We couldn't see culturable Borrelia, which is not surprising when that was expected. So what would be nice would be to repeat that study, but then do the 12 months time point or the 18 months time point when actually Barthold had seen by PCR and EMEA had seen by PCR those to become positive. That's really the time point that one would have to look at.
Just to follow up on that, what else is known about VBNC, the VBNC state, is that the type of stressor can have a different outcome, and there's a different window of time for the ability of the pathogens to resuscitate based on the stressor type, the pathogen itself, and so I think there's definitely going to be some variability, and I think, you know, the mechanism of the antibiotic is definitely represents a different stressor, and so we're not surprised to see different outcomes.
All right, question for Dr. Baumgarth. Have you quantified the SCD40 and SCD40L levels in serum of infected mice, or in immunized versus infected mice?
No, we have not done that. It's a good idea.
Okay. That was a shorter answer than I was hoping for, so I can find my...
I can expand on that.
No, it's okay. Yeah. Okay, so I don't know who this was targeted at, this particular question, but I'll pass it to Dr. Zhang because I think it may be relevant. If tick salivary glands have been shown to have biofilm like Borrelia? Or I don't know.
Yes, I think so. There have been studies done. Fred from New York Medical College, they've done some tick work and show there's a different type of persisters being present in the ticks. I think that's, there have been studies. He has a paper called Sleeper Cells. I think many of you probably have seen that. It's mentioned in that review article. Yes.
Dr. Baumgarth, could you or someone isolate from bacteria and identify a factor that downregulates BCL6 and CD45 T cells?
Yeah, that would be awesome.
Yeah. Yeah.
So the biggest, to me, one of the biggest hurdles of this kind of analysis work is that we can't grow Borrelia in culture and have them look like Borrelia in the mammalian host. So just having the amount of Borrelia that you need to isolate individual proteins to then see whether they have an effect. So you can go the recombinant protein approach where you just express recombinant proteins and see what effect they have. So, there are 8,500, I think, open breeding frames of Borrelia burgdorferi. So it would require a very high throughput approach to do that, and I'm not aware of anybody having that capability right now. So that would be, but in a way we have been thinking about how one can approach starting that, because you can't, Salmonella, you have a system to make mutants very easily, and you can just screen those. And Borrelia just isn't one of those bacteria that allows you to do that. So definitely the knockout route is not a way to ever go. So using recombinant proteins might be a way to do it.
All right, I think that's a good question to end on, because I think that's, maybe there's some collaborators out there that have some ideas on how to do something like that. So we should give another round of applause to the morning speakers. And excellent, and almost stayed on time to